How can a frog freeze solid and then hop away as if nothing happened? That's the real-life magic of the wood frog, a small amphibian that survives brutal Arctic winters by entering a state of biological suspended animation. This phenomenon, sometimes called biohack freezing, offers a glimpse into nature's most extreme survival strategies and hints at future possibilities for human medicine and space travel. Unlike most animals that would die from ice crystal damage, wood frogs tolerate being 70% frozen, with no heartbeat, no brain activity, and no breathing, then thaw out and come back to life in spring. How? They bioengineer themselves with antifreeze-like compounds, mainly glucose and urea, which protect their cells during the freeze. So what can humans learn from this frosty trick? These frogs don't just avoid ice damage, they actively control where and how ice forms in their bodies, keeping vital organs safe while ice builds in spaces between cells. This natural cryopreservation system inspires research into organ preservation, hypothermic surgeries, and even long-term space travel. Analogy, it's like pausing a computer mid-operation, freezing the screen, and then rebooting it days later without losing a single file. Biohack freezing challenges what we thought was possible in biology, showing that with the right biochemical tools, even death-like states can be reversible and life can be paused and restarted. How can wood frogs, Rana sylvatica, endure the freezing of up to 70% of their body water, yet revive unscathed when temperatures rise? And what strategies can we glean for engineering extreme cold tolerance in other organisms? Wood frogs orchestrate a suite of biochemical and physiological adaptations: controlled ice nucleation, massive cryoprotectant deployment, metabolic arrest, and repair pathways to survive winter's deep freeze. So far, so good. Until we recognize that freezing tolerance is not a single predictable switch, but a dynamic sequence of interdependent stages that inscribe memories of early cold exposure into the frog's tissues. Initial extracellular ice formation and rapid glucose mobilization set the stage for cellular protection. However, slight variations in cooling rate, cryoprotectant distribution, or antioxidant provisioning can dramatically alter survival outcomes, making the process far more complex than simple thermal suspension. At its core, three parallel adaptive processes drive, freeze, survival. Controlled ice nucleation, specialized blood proteins and membrane-bound nucleators, direct ice formation to extracellular spaces first, preventing lethal intracellular crystallization. Cryoprotectant surge. Within minutes of ice sensing, the frog releases enormous concentrations of glucose into its bloodstream and cells, lowering freezing points and stabilizing proteins and membranes, without incurring the toxicity that synthetic cryoprotectants would cause in mammals. Metabolic arrest and defense. As heart and lung activity cease, cells switch into ischemia-resistant modes. Antioxidant systems, glutathione catalase, and heat shock proteins surge to neutralize reactive oxygen species generated during thaw and to stabilize macromolecules. Yet even this mechanistic view conceals finer-scale intricacies. Different tissues deploy cryoprotectants unevenly. Brain and heart maintain higher glucose levels than muscle to safeguard critical functions. Epigenetic regulators prime gene expression weeks in advance, tuning enzyme levels and membrane lipid composition according to seasonal cues. Minor deviations in environmental humidity or pre-freeze hydration can skew the balance of ice distribution, leading to patchy cellular dehydration and uneven survival. At even finer scales, molecular chaperones and repair pathways add textural nuance to the freeze-thaw story. Freeze-induced protein denaturation is reversed by ATP-dependent chaperonins during rewarming, while DNA repair enzymes correct strand breaks incurred under anoxic conditions. Membrane phospholipids, enriched in unsaturated fatty acids, prevent phase transitions that would otherwise compromise bilayer integrity. Temperature oscillations during thaw trigger transient calcium influxes that activate autophagy, clearing damaged organelles before full metabolic resumption. In essence, biohack freezing is not a matter of plummeting to sub-zero temperatures and thawing out. It is a richly orchestrated ballet of anticipation, protection, and recovery. Each phase imprints chemical and physiological signatures that steer subsequent stages, making successful freeze tolerance a singular testament to evolutionary ingenuity. As with other natural paradoxes, from hysteresis loops to quantum curiosities, the wood frog's strategy challenges us to embrace multi-scale complexity if we hope to freeze and revive complex organisms without harm. Fundamentals of Biohack Freezing explores how certain animals, notably wood frogs, Rana sylvatica, survive deep internal freezing by leveraging extraordinary biochemical adaptations. These frogs can endure up to 70% of their body water turning to ice, entering a state of suspended animation where breathing and heartbeats cease entirely. Despite widespread ice crystallization, their cells remain structurally intact and biologically viable, resuming normal function upon thawing. Wood frogs achieve this by accumulating massive quantities of glucose in their blood within minutes of freezing onset, which acts as a cryoprotectant distributed across all tissues. It is like embedding antifreeze into every pipe of a house to prevent bursting when the temperature plummets. Unlike freeze-avoidant species that rely on supercooling or behavioral insulation to prevent freezing, freeze-tolerant organisms such as the wood frog allow controlled ice formation but mitigate damage through biochemical means. They produce cryoprotectants and proteins that manage ice growth spatially, distinguishing them from species like gray tree frogs, which avoid freezing altogether. This bifurcation reveals two fundamentally different strategies for winter survival: insulating versus enduring. It is like choosing between building a heated greenhouse to protect crops or genetically modifying plants to tolerate frost. At the microscopic level, the frog's liver rapidly converts glycogen into glucose upon sensing ice nucleation, flooding the bloodstream with concentrations exceeding 200 ml. This surge stabilizes cellular proteins and membranes, creating a protective environment that prevents intracellular ice damage. Additional osmolytes like glycerol support osmotic balance and membrane integrity. Within 30 minutes, a frog's tissues are saturated with glucose, effectively shielding them from freeze-induced injury. It is like instantly infusing a city's water pipes with non-toxic antifreeze to prevent blockages throughout an entire urban network. These animals also produce a suite of ice-nucleating proteins that orchestrate where ice forms, specifically in extracellular compartments, triggering solidification at higher sub-zero temperatures to avoid spontaneous and damaging intracellular crystallization. Simultaneously, antifreeze proteins near cell membranes suppress uncontrolled ice growth, tightly regulating spatial ice dynamics. For example, nucleators in the frog's skin initiate freezing at -2 DGC, giving tissues time to respond. It is like installing designated snow collapse zones around a house so that ice builds up only in structurally safe areas. Before and during freezing, wood frogs upregulate genes for protective enzymes such as superoxide dismutase, catalase, and various heat shock proteins that reduce oxidative and structural damage. These molecular repair systems activate even before full freezing, priming tissues to recover quickly upon thawing. This genetic preparedness ensures rapid repair of DNA and proteins damaged by freezing and thawing cycles. For instance, antioxidant enzyme levels spike during the frozen period to mitigate reperfusion injury during warming. It is like deploying a maintenance crew in advance of a storm to repair downed lines the moment conditions stabilize. Despite these defenses, the system operates within sharp survival thresholds. A slight increase in ice volume or a minor shortfall in cryoprotectant levels can transform a reversible freeze into irreversible cellular collapse. The coordination of cryoprotectants, membrane phase behavior, and ice-regulating proteins creates a non-linear survival boundary. Experiments show that deviating from the optimal cooling rate by even 1 dg/min drastically reduces viability. It is like walking on a frozen lake where staying just below a critical weight keeps you safe, but the smallest additional load breaks through the ice. These natural adaptations offer powerful templates for improving cryopreservation techniques in biomedicine. By mimicking the frog's rapid cryoprotectant delivery and spatially controlled ice nucleation, researchers aim to better preserve human organs for transplantation and create stable, long-term cell storage systems. For example, protocols that rapidly infuse glucose into mammalian kidney slices are being tested to reduce freezing damage. It is like applying a frost rescue kit to delicate flowers before shipping them through frigid environments, ensuring they bloom upon arrival. However, translating these strategies to humans presents challenges. Human cells differ in membrane composition, tissue perfusion, and cryoprotectant tolerance. High concentrations of glucose or glycerol can be cytotoxic, and even small deviations can cause osmotic imbalance. Moreover, ethical considerations arise around the potential for long-term suspended animation in humans, including issues of consent, personhood, and revival. Attempts to freeze human blood vessels using glucose often result in damage without adequate membrane stabilization. It is like trying to apply automotive antifreeze to a marine cooling system. It needs system-specific tuning to avoid harm. Looking ahead, new tools are emerging to bridge these gaps. Nanocarrier systems may deliver antifreeze peptides and cryoprotectants directly to vulnerable cell compartments, minimizing toxicity. Synthetic biology could enable temporary expression of frog-like freeze tolerance genes in human tissues during preservation. Advances in molecular imaging and real-time ice tracking will further refine freezing protocols. Early successes include liposomal delivery of antifreeze proteins that reduce damage in human hepatocytes. It is like upgrading an ordinary freezer with intelligent sensors and targeted defrosting to keep perishable goods perfectly intact through extreme conditions.